So far, we've explored sections 8.1 through 8.4 of your chapter on cell communication. And now we're going to talk about section 8.5, which is on enzyme-linked receptors. By the time we're done, you should be able to understand what is happening at the cell surface for our enzyme-linked receptors. You should be able to understand once those receptors are activated, what is the role of the relay proteins and how they eventually lead to a protein kinase cascade. What exactly does that mean? And how is it that they ultimately lead to the activation of specific transcription factors that drive gene expression? All right, so just to remind you, we are talking about enzyme-linked receptors. So these are going to be cell surface receptors that have enzymatic or catalytic activity. So just to orient you on the slide, this would be our extracellular space, this would be the intracellular space, and these gray rectangles are our phospholipid bilayer. So within the phospholipid bilayer are nested these cell surface receptors with that extracellular domain that's going to be responding to the signal that is going to have the catalytic or enzymatic activity. So what we see here is a particular cell surface receptor. This is called RTK, or receptor tyrosine kinases, and I'll explain that in a bit more detail on the next slide. But these are going to be two identical portions to one another that are not interacting currently in the unbound state. They're not interacting with each other. However, upon the binding of ligand that you can see here, once those ligands bind, the two monomers are going to dimerize. They're going to come together, and then what you see is once they come together, they're going to actually phosphorylate one another. This is called autophosphorylation. And so now this is our activated receptor tyrosine kinase. These phosphorylation events took place specifically at tyrosines that were in the particular receptor. Once you have ligand binding and phosphorylation has taken place, we're then going to recruit different types of proteins that are in that intracellular space, and that will lead to a signaling cascade. All right, so before we get into the specifics of this pathway, I wanted to remind you about phosphorylation. So when we talk about having kinase activity, that means that a phosphate group is going to be attached to a particular amino acid on a protein. And there are only, there's just a few amino acids that actually can get phosphorylated. So serines can get phosphorylated, threonines can get phosphorylated, as well as tyrosines. So in the pathway I was just talking about here, this is called receptor tyrosine kinases. So within this particular protein, there are several tyrosines that are going to get phosphorylated, and once phosphorylated, then the receptor is deemed activated and has that catalytic activity. All right, so let's look at a specific cell signaling pathway that would fall within this subclass. All right, so we have a particular cell surface receptor called epidermal growth factor receptor. Just to take a step back, if you think about what comprises your epidermis, those are the cells that are on the outside of your skin. So if you think about when you have a wound or a cut on your skin, eventually you know that skin, if the cut is not too deep, will repair and you'll be able to regenerate some skin cells, and in a week or so it won't appear that you have a cut anymore. Well, how is it that that's actually taking place? So for a particular cell, it is going to be receiving a signal that repair or cell division ultimately needs to take place. And so that is what we are showing here in this cell signaling pathway. So epidermal growth factor is going to be our ligand in this scenario, and our receptor is epidermal growth factor receptor. So just like I showed you in this slide here, without the presence of the ligand, our receptor would have been undimerized. However, upon ligand binding, these two receptors are going to find each other in the phospholipid bilayer, and they're going to start to phosphorylate one another and basically initiate this cell signaling pathway. So once phosphorylation has taken place, there's going to be recruitment of this protein called GRB2. And GRB2 will interact with the phosphorylated form of EGF receptor. Once it interacts with the receptor, the activated receptor, GRB2 will undergo a conformational change. Once GRB2 undergoes a conformational change, it's then going to be able to recruit SOS. And SOS, once interacting with GRB, is going to undergo a conformational change and will interact with another protein. This is called a lipid-anchored protein named RAS. And what you'll see here is that RAS can interact with GDP, guanosine diphosphate. So this is also another word for this is a G protein. And upon binding with SOS, RAS will dissociate from GDP and will end up interacting with GTP, guanosine triphosphate. And the interaction of RAS with GTP will allow for the recruitment of another protein target called RAF. And once RAF is recruited to RAS, that's going to initiate a phosphorylation cascade event where we can see a downstream target named MEK is going to get phosphorylated. So MEK in its unactivated state would not be phosphorylated, but now you can see MEK is phosphorylated. And MEK is going to lead to the activation and phosphorylation of ERK. Here you can see ERK now gets phosphorylated. ERK is then going to be able to go from the cytosol into the nucleus itself and is going to activate different transcription factors such as MYC and FOS. And MYC and FOS can actually bind in the promoter regions of DNA and turn on gene expression. They're going to turn on certain genes that eventually will lead to the synthesis of mRNA. The mRNA will then get translated into proteins. And now we are going to have the specific proteins that are necessary to drive cell division, telling this cell to go ahead and replicate itself because, initially the signal that we had received was that there had been damage to our cells and that we need to go ahead and replicate this entire cell. The end result is going to be proliferation of this cell. This healthy cell will be replacing the damaged cell that had been the result of injury. All right, I just want to make clear when I'm talking about a phosphorylation cascade, what that means is that you have an initial protein that has this kinase activity, again, the ability to phosphorylate, and it's going to transfer a phosphate group to an inactive protein and rendering that protein active. And this now downstream target, which is now phosphorylated, is going to be able to activate a different downstream target. And this downstream target will initiate another downstream target. It's kind of like a game of telephone where the message initially received is going to be transferred from one to the other until the cellular change that was desired. In this case, we want to make a particular protein, so we need to turn on the expression of a particular gene is going to be relayed to the right target. And this phosphorylation, as I described in the previous slide, led to turning on a certain set of genes, but this phosphorylation cascade could also be responsible for turning off particular genes. The reason that we do this is you get a major amplification of your initial signal. And so by our initial epidermal growth factor that was received, once we initiate this phosphorylation cascade, we're going to be able to amplify and relay that response to drive the correct cellular response. Okay, so at this point, I would put the video on pause, and I would like for you to be able to explain this to yourself or to a family member, or if you're with one of your colleagues, explain it to one of your colleagues. All right, so it's really important that we're able to induce these pathways when we need them. So in the example that I just had given you, we were wanting to, if the cell had undergone cell damage and we needed to repair the damaged tissue, then you were going to take a healthy cell and go ahead and cause it to divide. Now, eventually, you're going to want to inhibit that process because you don't want your cells to constantly be dividing. We would have a massive growth on the outside of our skin. So how is it that we actually are able to turn these pathways off? Well, the key player here is really that RAS protein. If I go back a slide, when we are able to take this RAS that's interacting with GTP, if you can hydrolyze that and get rid of a phosphate group, such that it's now interacting with GDP, guanosine diphosphate, RAS is rendered inactive and the whole pathway, the signaling cascade, will stop. So one question I have for you is, if a mutation resulted in the RAS protein that normally is able to hydrolyze that GTP to form GDP and inorganic phosphate, but if it could no longer do that, how would that affect this signal transduction pathway? So again, if we were not able to hydrolyze the GTP and we consistently had activation of this pathway, what would be the outcome? So hopefully you are beginning to come up with some ideas about that, but for the answer to that question, I would like for you to watch a video that I'm going to link right under this video that I'll be posting for you that kind of directs you at the correct answer. And I'll have a question in your follow-up quiz regarding this specific question. All right, thank you so much.